Hello and welcome to another very entertaining video. This one will complete our series on macromolecules. We've talked about carbs, we've talked about lipids. We're not going to talk about nucleic acids right now. We're going to focus now on proteins, and that will end this unit. So let's see if we can follow the same pattern that we tried to use on the others, where we identified monomers, the building blocks, and then we actually built these things. And this is arguably more important for proteins than it is for the others, simply because if there's one thing we have to know, it's that protein function is derived simply by its shape. The reason any protein does a specific thing is because of the shape of that protein. And so understanding shape or form is critical. We often say form is function when it comes to protein structure. And that's why we're going to highlight all this stuff over here, right? Primary structure, secondary structure, tertiary structure, quaternary structure. We have to understand what these levels of structure are, because the shape of the protein is so critical to understanding how it works. Just to highlight that, we look at hemoglobin. Hemoglobin is actually 4 different protein subunits that interact with each other. Two of them are, you have 2 alpha and you have 2 beta, but here it shows you them, as 4 individual subunits: the red, the purple, the green, the blue. It's important that all of those things interact in that exact way in order for hemoglobin to function the way it does in carrying oxygen and carrying carbon dioxide. Hemoglobin is really a marvel when it comes to the little machine that it is. But it's critical that it's shaped just the right way. The other enzyme we have up here is lactase. Enzymes, the vast majority of enzymes are proteins. And recently talking about carbs, lactase is the enzyme in the brush border of the small intestine responsible for binding to lactose and breaking it apart into glucose and galactose. I hope you can appreciate lactase has just one job, and that's to bind to lactose and break it. The shape allows for that to happen. If the shape is modified just a little bit, then maybe it couldn't bind to the sugar. Maybe it couldn't bind to lactose. Or maybe it could bind, but it couldn't stress it enough to break that bond, right? Shape is so very important when it comes to proteins. And that's why we spend all of this time talking about primary, secondary, tertiary, and quaternary structure. Okay, so let's get into primary structure. Primary structure is simply defined as the order in which amino acids are connected. So within this definition, there's a couple of things we have to look at. Number one, amino acids, right? These are the monomers, the building blocks of protein. So let's take a second to look at an amino acid, see what it looks like. Going back to our little video on functional groups, hopefully it's a good reminder. We can see why this is called amino acid. It's got an amino functional group, it's got a carboxyl functional group, which acts as the acid, and from that we can go on to understand how amino acids play with each other. So let's start by drawing our amino group. These are hydrogens here, and then we've got our alpha carbon. And there we've got our carboxyl group. And of course, off this alpha carbon, we need a hydrogen there. And then we have what's called the R group, or the side chain, right? And this R group is what makes amino acids different. There are 20 basic amino acids that we use for building proteins. There's more amino acids, there are other modifications we can make, but there are 20 basic amino acids that we can build for proteins. About half of these are essential, meaning we have to eat them in order to have them present in our body. The other half we can make usually from sugar, so we can turn carbs into glucose in specific, turn this into various amino acids. So, but half of them we actually have to eat, and that's why people could become protein deficient. It would be very hard, unless there's some weird specific genetic disorder, it would be very hard to be deficient in any one amino acid. because of how all of these things come when we consume protein. But it is possible to become protein deficient, and arguably a majority of Americans are not getting enough protein in their diet. But when we think about that, what does it mean to eat protein, right? We're eating these massive pieces of protein that are made of these 20 different building blocks. So let's first show you how to assemble these. Let's draw another amino acid. And we're going to follow the same pattern that we follow both for lipids and for carbs. We're going to glue these things together simply by dehydration. So we identify water. The dehydration synthesis will remove that water. H2O comes out. And now the carbon of the carboxyl group on the first amino acid is going to be directly attached to the nitrogen on the second amino acid via covalent bond. And all of this happened because of the removal of water, dehydration synthesis, right? And the resulting bond, right, this bond right here between the two amino acids is called a peptide bond or a peptide linkage. And as we add more amino acids, another one and another one and another one, we start to build a polypeptide chain. And the order in which we put these defines our primary structure. So now the next question is, well, how does the body know in what order to put the amino acids? That's a great question, and that comes back to the central dogma of biology. So let's talk about that for just a second. I don't want to spend a lot of time on it, but the central dogma of biology can best be described like this. DNA can be copied or transcribed into RNA, mRNA in particular, and this RNA is then translated into protein. So what does this tell us? It tells us that the code to know in what order to assemble the amino acids is buried inside of our DNA. In fact, that's what DNA is. All the DNA in your cells simply is a code to tell the body how to assemble proteins. And I do want you to appreciate the diversity that this is, right? If I had 20 different colors of beads, I'm not going to draw 20, we'll just draw a few, but think about this. We've got some blue ones, maybe some orange ones, throw in some red ones, some green ones. So I've got 14 different beads down there, 7 different colors. If I strung these up, what are the odds that I would put those in the exact same color sequence as you would? Or if I did it twice, right? What are the odds of doing that? I hope you can appreciate the diversity quickly that we can build in this string of beads, simply here showing 7 different colors. Of course, with amino acids, we've got 20 different amino acids, and these are going to be anywhere from, on the short end for a protein, 50 to 100, that's a little bit short, but on the long end, several thousand amino acids long. How do we know in what order to assemble them? It all comes from the DNA. So what does this look like? If this is an introduction to you for DNA and RNA for the first time, DNA is just made up of 4 letters, right? It's made up of a ribose sugar, it's got a nitrogenous base attached to it, and some phosphate. But the easiest way to think about it, right, is we've got adenosine, or thymine, or guanine, or cytosine. These 4 different building blocks of DNA. RNA is going to be very similar. The only difference is instead of T's, we're going to use uracils. So for RNA, we have A, U, and G, and C. In no particular order, because it's the order of this in which we actually learn how to assemble proteins. Now, we read this code actually in triplets, 3 letters at a time, in what we call a triplet codon. So let's just randomly make up a series of letters. We're going to use mRNA. So maybe it's A, U, C, C, G, U, A, C, G, A, U, Now, what I want to do is I want to divide this up into threes. AUC, CGU, ACG, AUA. Now you can just quickly Google something called the triplet codon code because we know how to interpret this. So I look at that code and I can see that AUC actually codes for the amino acid isoleucine. CGU, the next one, arginine. ACG, threonine. And AUA, another isoleucine. That's the abbreviation. Let's add one more. Let's add onto this UAG. Well, this one is what we call a stop codon. That tells the machinery it's time to stop. We're done translating this protein. The machinery, the cellular machinery that works on this is a little organelle called a ribosome. And the ribosome kind of comes in two parts, a slightly bigger part and a slightly smaller part. We don't need to know the specifics of that, but the ribosome is what's going to translate or interpret this code. So as this mRNA feeds through this organelle, of course we can appreciate the first thing that's going to come in is this first codon, this AUC, right? AUC is here. The body has another type of RNA called a tRNA, and it's going to read just the opposite. A's always bind with T's, or in this case U's because we're talking about RNA, and C's always bind with G's. So along comes this tRNA, and the tRNA has a sequence that recognizes that triplet codon. The tRNA will read UAG, right? It's the complement of that sequence. And of course, this tRNA is attached to the specific amino acid isoleucine. So the tRNA specifically bound to isoleucine comes because it's recruited to that code. That's how I know that isoleucine is going to go in there. Of course, this whole thing shifts over. So let's shift it really fast. And the next one in is my CGU. The complement of that, C's always bind with G's, right? So we've got a G, C, A, and this one is going to be bound to the amino acid arginine. And then the ribosome has catalytic activity. It's going to take these two amino acids right here, and it's going to fuse them together, the peptide bond, the dehydration synthesis reaction that we just showed you, and this is how I start to grow my chain. So the code allows us to know the sequence. As the code enters the ribosome interprets it in groups of three called triplet codons, and that's what recruits the proper amino acids so that I can build the correct polypeptide chain. So now coming back to primary structure, the order in which amino acids are connected, right? For every protein, the order is slightly different. The purpose for that different order will become more obvious as we talk about the other ones. But when we talk about primary structure, we're talking about that order. And I would make note, right? Many genetic diseases, if I've got a mutation in the DNA, that has the potential to change the code. And if the code is changed, then I'm going to put in a different amino acid. Or maybe it gets mutated to a stop codon, and then I just stop when I'm way too early to stop, and so the protein becomes truncated. If I change that amino acid, and it's kind of in a sensitive, important part of that protein, maybe it alters the way that protein functions. And this is how genetic diseases arise. By changing the DNA, I change the primary structure, and that potentially could change protein function. All right, with primary structure done, let's move on to secondary structure. Secondary structure probably has the most stringent definition. Secondary structure is the formation of either an alpha helix or a beta pleated sheet stabilized by hydrogen bonds, specifically along the backbone of the protein. So let's go back and let's look at our growing polypeptide chain and see if we can identify what's going on here. So to understand this, let's first define backbone. Right? We've got our basic amino acid that we've drawn over here. We went through all the parts of it. As we string all these together, we've got this growing polypeptide chain. There's some of it that I haven't drawn because what we really want to focus on is the backbone. Every single protein in the body follows this order. NCC, new amino acid. NCC, new amino acid. NCC, new amino acid. This is the backbone of the protein. We are not, and this is the important part of the definition, we are not taking into consideration interactions with these side chains, these R groups on the alpha carbon. We're only thinking about the backbone right now. Okay, so now we got to think about hydrogen bonds. If you recall from talking about water, hydrogen bonds occur as an attraction between two electronegative elements that bridge a hydrogen. In other words, I get bonding between partial charges associated with polar covalent bonds. Where along this backbone can I see polar covalent bonds that might allow this to happen? Well, let's look at this carbon right here. This carbon is double bound to oxygen. Those are polar covalent bonds, and that's going to put a significant partial negative on this oxygen. In contrast, if we look at the nitrogen bound to a hydrogen, this bond right here is also a polar covalent bond. But because nitrogen is more electronegative than hydrogen, that puts a partial positive on that hydrogen. So now we can see how these hydrogen bonds are going to form, and it's going to be along the backbone. Now, what I want you to appreciate is that the alpha helix that is formed, if we take this polypeptide chain and for a section of it, right, not for the whole thing, but for a section of it, let's twist it up, just like an old school telephone cord. This is the alpha helix. And it's stabilized by hydrogen bonds along the backbone. That's the idea, that these interactions between the electronegative elements of oxygen and nitrogen that bridge that hydrogen bound to the nitrogen, this is what's stabilizing this structure. Of course, a beta sheet's slightly different, where for a section of it, maybe I'll come up and, maybe there's a bunch of stuff going on up here, but then I'm going to come back and I'm going to run, in this case, anti-parallel, or maybe I run parallel, and then we swing back, and then we go on and we do some other stuff. Same idea, where hydrogen bonds between these strands as they run parallel or anti-parallel to each other stabilize that specific structure. So here we have the alpha helix. Here we have the beta sheet. These are the two secondary structures stabilized by hydrogen bonds. Now we can actually see that if we go back up and look at the models that we were looking at before. Everything that you see, let's look at lactase, everything that you see here in purple, those are all showing these alpha helices. You can also see, we see this arrow right here and the ribbon structures like this. These are the beta sheets that we're looking at. Not every protein has alpha helices. Not every protein has beta sheets. Some have lots and some have a few. It just depends, again, on the primary structure. That's what dictates all of this stuff. We look at hemoglobin. No beta sheets in hemoglobin, but lots and lots and lots of alpha helices, and that's what contributes to its overall function. So, by definition, secondary structure, an alpha helix or a beta sheet stabilized by hydrogen bonds along the backbone. All right, let's transition now to tertiary structure. In our textbook definition for tertiary structure, the three-dimensional shape of the protein stabilized by all types of bonds that we've talked about anyway. But here's the unique feature. These bonds are found between the R groups, or the side chains. So when we think about all types of bonds, right, we can list these. We've got certainly ionic. We have the ability to make covalent bonds. These are going to be disulfide bridges. Certainly hydrogen bonds. An important one would be hydrophobic collapse, where all the hydrophobic R groups are going to find each other and collapse to the middle because they're trying to avoid water, right? Most of this is happening in water. So these are all contributing to the final three-dimensional shape defined as tertiary structure. Now I do want to just give you an idea of how these R groups are contributing. So let's draw a few amino acids. All right, on the left we've drawn lysine. On the right we've drawn glutamic acid. Please appreciate once again everything in white. This is the backbone, right? That same repeating NCC. And we can see the amino group on the left. We can see the carboxyl group on the right. This is how we're going to attach amino acids to each other through the peptide bond. That's what gives us our primary sequence. And also interactions through this backbone gives us our primary structure. Interactions via hydrogen bonding gives us our secondary structure, the alpha helix, the beta pleated sheet. But now we're talking about these side chains. And we can see the differences, lysine, glutamic acid. What I want you to recognize is, going back to the idea of functional groups that we talked about in a previous video, we look at lysine. And the functional group, right, we're looking down here. This is an amino group. And we know that nitrogen, because it has this lone pair of electrons right here, is a great docking site for protons. So oftentimes you'll see it drawn where people have the H there and a positive sign. So because it acts as an organic base, it's a proton acceptor, and as that proton comes, it ionizes it. We charge it, so it's got a plus charge. I hope you can see where we're going, because if we look at glutamic acid, we also have a functional group. But this one is the carboxyl group, and if you recall, the superpower of the carboxyl group, because I've got two oxygens on that same carbon, collectively the electronegative power of oxygen is pulling those electrons off of the hydrogen, and this hydrogen is actually going to leave. So it pops off, and that leaves behind a negative charge. So now I've got an amino acid with a positive charge and an amino acid with a negative charge. If these things are oriented close to each other, what do I get? I get an ionic bond, right? One of the bonds up here that's stabilizing this three-dimensional shape. And so this is just an example of how two different amino acids, two different side chains, can interact with each other to stabilize the overall three-dimensional shape of the protein. All right, last but not least, quaternary structure. This is simply defined by multiple subunits interacting with each other to create a functional structure, right? We saw that above in hemoglobin, where I actually had 4 unique subunits, 2 alpha and 2 beta, that were all playing with each other. That's simply the idea. There's not a whole lot more to describe here. Some proteins need quaternary structure. Other proteins, like lactase, shown above, were completely independent of other subunits, and so they don't need and don't have quaternary structure. The interactions, again, are all types of bonds that we've talked about, and they're occurring between R groups, and it's all just stabilized by these interactions. Now, sometimes the protein or the quaternary structure, there might be 2 subunits. Sometimes there might be dozens of subunits, something like RNA polymerase, fairly complicated. There could be hundreds of subunits kind of coming together to make sure that whatever this protein macro complex is, it's functional, because there are a lot of things that are driving it. And I hope you do take the time to appreciate the beauty of how all of these proteins had to co-evolve, had to evolve together to create that structure. It's mind-boggling how that was even able to happen. Even something as simple as, if we go back up to hemoglobin on the top, right, here's hemoglobin. anyone that studied hemoglobin, how it can change its affinity for oxygen based on the presence of increased acidity or temperature or metabolic waste products like 2,3-BPG, all of these things actually interact with hemoglobin and allow hemoglobin to deliver oxygen to the tissues in the body that need it most. And it's all because of what hemoglobin looks like, its shape. And the fact that we've got these 4 different subunits that are all functioning to make that happen, it's mind-boggling how that could actually occur through evolution. A really, really amazing thing to think about. So anyways, as we look at this now, remember, for proteins, form equals function. It's all about the shape of this structure. And to get to understand where that shape comes from, we divided it up into primary, secondary, tertiary, and quaternary structure, each with its own textbook definition and each helping us understand how to get to that final 3-dimensional product.